I'm very happy today to be joined by Dr. James Doroshow. He is Deputy Director for Clinical and Translational Research at the National Cancer Institute, and he's giving a talk which is called New Paradigms for Early Phase Clinical Trials. Thank you so much for being here.
It's a pleasure.
You're the leader of the NCI's Clinical Trials Working Group. Would you begin by describing how you're restructuring and reforming the clinical trials process?
So we've been actively engaged actually over the last 7 years at looking at all parts of the NCI's clinical trials activities. The most recent, which culminated actually this past Monday with approval by the NCI's Board of Scientific Advisors for a new RFA that will support a completely reconfigured NCI clinical trials network. It focuses our efforts into 4 adult and 1 pediatric clinical trials group that will do both late-stage phase 2 and phase 3 investigations as a network rather than as separate individual oncology clinical trials activities in a way that really has never been done before. And so we're very pleased that that has actually come to fruition.
Would you discuss phase 0 trials and how the need for this came about?
So also about 5-6 years ago, it became clear that we, both in the oncology community and also with the help of our colleagues at the FDA, wanted to figure out a way how to do pilot trials to get information on both bioavailability, on whether or not a compound to hit its particular target, on whether or not we could do very early imaging studies with less toxicology and other very expensive preclinical information that is normally required for a standard IND. And so the exploratory IND was born out of a collaboration between the FDA and the NCI that actually allows investigators to do single-dose studies, either microdose or doses of drugs that are unlikely to have toxicity but are likely to hit their target, so that if you have the appropriate assays, whether they're imaging assays or biochemical assays, one can determine very early in the drug development process. Actually, one can compare a series of different analogs to see which one is both absorbed most readily orally, or which one of a series might be most effective in hitting its molecular target. And this can be done in such a way that it's actually less expensive and faster.
Are there additional foci for the phase 0 trials intended beyond targeted therapies?
Well, so there's a great deal of interest actually in using this approach, and a very appropriate approach for cancer prevention. We often are faced with a situation in which we have a variety of different compounds. We don't know which ones of those are well absorbed. We don't know whether actually they will hit their target in preneoplastic lesions. And so earlier this year, one of the first, probably the only earliest phase 0 trial in cancer prevention was published in Cancer Prevention Research with the idea that there were a series of relatively non-toxic substances that were likely to have a particular endpoint early in the development of a cancer, but it was unknown whether those compounds would be well absorbed. And so over a period of less than 2 months, investigators at the Mayo Clinic were able to discern which of those compounds to bring forward to larger cancer prevention trials.
Now, big picture, how do you envision the clinical trials process in the future?
I think as we know more tumor biology and as our ability to measure particular molecular characteristics of tumors enhances, as we're seeing at this particular meeting, we'll be able to do actually smaller trials with more information, with bigger differences between standards of current standards and new therapies that will actually allow us to make the drug development process much faster.
Thank you, Dr. Doroshow.
My pleasure.